you, Jose, thank you so much. This is a terrific symposium. I learned so much. And as you heard from, so thank you for the invitation. It's really an honor to be here. As you heard from Peter Byers, it's really tough to come to San Diego from the East Coast. Cold and so beautiful. The sunshine is just gorgeous. And by the way, so his remark about first cousins brought back a memory which I completely forgot, obviously. So I had one first cousin. She was really beautiful, and my expression must have been so obvious that I remember distinctly my grandmother, my mom's mom. One day she just blew up and she goes, "Just stop it! Just marry her." So, Italians are not very subtle. We're not very subtle people. So we just anyway. So I'm going to tell you about the MHE, multiple hereditary astrostosis, and I'm going to talk about the pathogenic mechanism in mice. So unfortunately, we don't have anything in humans, unfortunately, as yet. And I will leave the prospect for therapies only if you ask me. So I'm not going to venture too much into that. So MHE, which is also known as MO, multiple histochondroma, or HME, same disease, autosomal dominant, the frequency is about 1 in 50,000, is diagnosed at birth or early postnatal life, so it's considered a birth defect by definition. And it's characterized by this exostosis, which are benign tumors, but also several other problems, including chronic pain, skeletal deformations, wound healing problems. And you can see the exostosis sticking out here from the long bones. They can be in the pelvis, they can be in the ribs, they can be in the spine. And they are benign, but unfortunately they transition to malignancy, about 2 to 3% in the patients. They become osteosarcomas, and then things are really, they can be life-threatening, and often they are. So why are we interested in this? Not only for the disease, but there is the biology behind, and that's what we are trying to understand, the biology behind it, the pathogenesis, and try to figure out whether there is anything we could do to stop this disease, and in particular the exostosis. So they are cartilaginous outgrowths, and they form next to, but never within, the growth plates. And some benign tumors or cartilage actually form within the growth plate. This exostosis formed next to, but never within. So there is a topography that we are trying to figure out. What's the regulation of the topography of this exostosis? What does it form here? And because they form here, they interrupt this magic border between the growth plate and the perichondrium, which is normally uninterrupted. So they stick out and interrupt that border. So this disease has been studied for many, many years. This is a review article from Jeff Esco, who is back there, one of my collaborators, also with Yu Yamaguchi. And there are about 1,000 mutations now known in these two genes, EXT1 and EXT2. They are all loss of function mutations. Most of them are nonsense, some are missense. And these two genes code for these two glycosyl transferases in the Golgi that co-polymerize heparan sulfate. And so therefore, since the patients are heterozygous for EXT1 and EXT2, they have a heparan sulfate deficiency throughout the body, probably about 50% of normal levels. So why is this a problem? Well, as in this review that also Jeff Esco published a few years ago, heparan sulfate regulates so many numerous physiological processes that when you have a deficiency, in theory, most of these processes could be affected. And the one that we are paying most attention is the fact that heparan sulfate proteoglycans bind to signaling proteins, absolutely key regulators of essentially anything, hedgehogs, BMPs, FGS, WNTs, and they regulate distribution and activity on target cells. So again, a deficiency in heparan sulfate could alter these processes, and this is where most of our work is now centered. So every time I give this talk, I change the question because there are so many. But today I'm going to try to focus on this one here and leave this for later. So how do heterozygous ESC mutation, HSC deficiency, lead to histostoses formation? So what's the mechanism? And what do histostoses form next to, but not within the growth plates? Regarding the first one, so when Stickens, Jeff, and Gina Werb produced these mice, they tried to mimic the human condition. They made just heterozygous mutant mice in XT1 and XT2, but to their surprise, the mice were largely normal. And only about 15% had developed an histostoses here and there, and these histostoses were only in the ribs, were often solitary, and depending on the genetic background of the mice. So they were really not mimicking MHE as yet with this, this. So what we did, again, collaboration with Jeff Esco, we said, okay, how about double hetero? Why did we say double hetero? Because since the two enzymes are required for HS synthesis, we predicted that the double heads will produce less HS sulfate than single heads, and this might actually mimic much more closely the human condition. And the answer was yes. In fact, these double heads really mimic very much MHE. They have multiple histostoses, they have in the ribs, many. They have really stereotypic histostoses in long bones, which is one of the most frequently site affected in the disease. And in fact, they have even two types of histostoses, which are similar to the one we see in humans, in patients, which is the typical histostosis, which is a growth plate-like structure, oriented at 90 degrees compared to the long axis. So the growth plate, the normal growth plate is in this direction. Histostosis always go in this direction, which is another puzzle. And also they have some histostoses that look like transition to malignancy. So we have really, and in fact, these double heads have many more histostoses than single heads, and the frequency is almost twice as much. So in this paper that Yu Yamaguchi published a couple of years ago, 2.5 years ago, what they did, they went on to the next step, which is they create conditional EXT ablation. And in fact, what they found, once you conditionally ablate the EXT1, not throughout the organism, otherwise the mouse would not exist. So mice cannot exist without the EXT1 and EXT2. So they had to be conditional. And in this case, they found 100% penetrance, as you can see in many places here. And this conditional knockout was done with COL2 CRE-ER and COL2 directs CRE activity in chondrocytes. And so, in part, we have begun to answer this first question: what's the relationship between these mutations and histostoses formation? Because clearly, at least in patients, appears to be the single heterozygous ES1 ES2 mutation in patients might not be sufficient to trigger multiple histostoses. And histostosis formation is inversely related to XT function. So the more XT function you lose, the more histostosis you're going to get. And, therefore, we are trying to figure out how this relates to the pathogenesis in patients. Let me tell you a little bit about this, why these histostoses grow next to the growth plate. And so our hypothesis was that since they form next to, but not within the growth plates, might be the pathogenesis of the histostoses involves perichondrium. And it might also involve ectopic signaling by prochondrogenic factors. So if there is prochondrogenic factor ectopic in perichondrium, perichondrium might be responsible, at least in part, for histostosis formation. And the answer to this question seems to be affirmative to both of them. So we conditionally eliminated the EST1 in the epithelial perichondrium using this transgenic mouse here, this GDFI CRE. And as you see by rosa staining, there is activity of CRE recombinase along the perichondrium located in the epiphysis. And sure enough, when we did that, histostosis began to appear. So this is the border between perichondrium and growth plate in the control mouse, perfect border, no interruptions. And in this knockout, the conditional knockout, as you can see, they begin to be these little this little exostosis, and a few days later they become much more apparent. And we were able to continue to understand what was behind this. And so in this experiment, these are EXT flox long bones from mice in a splan culture. And we ablated EXT by adenochree. So just adding adenochree to the medium, and this is the location of GFP. It shows you that the virus is all over the place. Sure enough, in this model, the border between perichondrium and growth plate is intact in the controls, but in the adenochree-treated specimens, as you can see, you begin to see ectopic formation of cartilage, which is an exostosis like tissue. And interestingly, even though the virus was all over the place, over the entire length of the bones, the formation of this exostosis-like tissue actually was mostly confined to the upper epiphysis. And we went to look both in vivo and in vitro in organ culture, whether there might be ectopic BMP signaling. Why BMP signaling? You heard from Eileen just a moment ago. BMP is a major prochondrogenic factor. So if there is an activation of ectopic cartilage formation, probably there is ectopic activation of BMP signaling, and the answer to this is also yes. So both in vivo and in vitro, the perichondrium in controls is negative of phosphoSMADs, which are the mediators of BMP signaling, but in the knockout, either in vivo or in organ culture with the adenochree, you see ectopic BMP signaling all along the perichondrium. And this change precedes by about 24 hours to 48 hours the formation of cartilage. Now, we actually, years ago, this was in 2007, we published this paper in which we were looking at Indian hedgehog, which is another major prochondrogenic factor. And what we found in this particular mouse model, which has not much to do with the MHE, but it was very, very informative, what we found was that, in these knockouts, the Indian hedgehog was relocated all along the perichondrium. So this is the distribution of Indian hedgehog in wild types. And in this mutant, it was Indian hedgehog proteins was all redistributed all along the perichondrium. This led to ectopic signaling of hedgehog because we were monitoring one of the targets of hedgehog, which is PACHIT1. And sure enough, a few days later, cartilage appears. Okay, so again, here's the border. This is the wild type, growth plate, perichondrium, perfectly intact. A few days later, after we activate this mutation, boom, we get ectopic cartilage formation following the redistribution of hedgehog. And we went on to just do this experiment in the last few months. And so what we did, just to confirm what I just showed you, we made this triple transgenic mouse. So this is EXT1 flox got to 3ER and GLI1-LAC-Z. GLI1-LAC-Z is a hedgehog signaling reported mouse line. And this allowed us to show that, in fact, the formation of this ectopic cartilage, as you can see here, this exostosis is accompanied by the redistribution of hedgehog, because this is, as you can see, there is very, very strong GLI1 activity here in the perichondrium in the mutant, but not in the wild types. So we got to this point, we began to do experiment in vitro to understand a little bit more the mechanics of these changes. And as you now are, it's obvious that what's happening in this disease is that if we inhibit heparan sulfate or we lose EXT1 expression, we actually gain chondrogenesis. So the loss of heparan sulfate, EXT1, is you get the opposite response. So what's the relation between this inhibition and the stimulation? So what we did was just take this mouse embryo limbud cells, and this is one of the most favorite chondrogenic system in the world. So you just take mesenchyme from limbud, from mouse embryos, you put them in vitro, and they undergo spontaneous chondrogenesis. So these embryos were EXT floxed, and what we were able, therefore, to do either treat them with adenocre to knock out EXT from expression, or we simply treated these cultures with heparanase, or we treated them with Sorfan, which is an HS antagonist. We just wanted to do 3 methods to see whether we would get the same results. So in all these situations, either we knock out the gene with adenocre, or we treat the cells with heparanase, which will destroy heparan sulfate, or we treat them with Sorfan, which is HS antagonist, the answer was always the same. We boost chondrogenesis. So once again, loss of heparan sulfate, loss of EXT gene expression, much more chondrogenesis. And this was accompanied by, obviously, all the genes for chondrogenesis that go up many folds. So we want a little bit more in detail to try to understand what was happening. So this is, just pay attention to this panel here. This is a reporter assay. So these are the same cells I showed you before, but transfected with a luciferase BMP signaling reporter plasmid. And what we found was, so this is the level in the control cells. I just have it here in bigger. So if you take, these are the control cells. So again, this is BMP signaling activity in the ordinate. So this is the level in the control cells. If we treat them with BMP2 from outside, obviously BMP signaling goes up, as you would predict. Interestingly, we got the same level of activity if we treat them with Sorfan. So we block HS activity, BMP signaling goes up as much as treating the cells with BMP2, and when we obviously combine these two, we get 20-fold increase in activity. So we also monitor BMP signaling, and if you look at p-phospho-SMADs, they go up very quickly as soon as the cells are treated with Sorfan, or we treat them in the other methods I told you before, either adenocre or heparanase. So the cells are very quickly responding to this loss of heparan sulfate function or EXT expression, and they increase chondrogenesis. And interestingly, not only they are signaling more, they're actually gene expressing more BMP2 and the 2 receptors for BMP. So everything is being boosted up by the cells in response to this loss of heparan sulfate or loss of EXT function. So I just have 2 or 3 more slides. So in 2 or 3 years ago, this paper came out, which really caught our attention. And I have to say, it has not been, as far as I know, this data have not been repeated by anybody. We are trying now to actually do it ourselves. So we're just collecting human specimens. And so what this group reported, that in these human MHE patients, if you just prepare sections of their cystosis, and you look for heparanase, endogenous heparanase, it's much more evident in the pathological tissue than control. Okay, in control, there is nothing. These controls are pieces of ribs. These are humans, so whatever the doctors gave them. And interesting, they took cells out of this MHE masses of tissue, obtained a surgery, and somehow they also got normal chondrocytes from also donors. And they claim that, in fact, they confirm what I just showed you before, that the MHE chondrocytes have much more heparanase than the control cells. This is endogenous enzyme. And so Julianne, who's doing all this work with us, she actually tried to do this, tried to figure out whether this is the same in our system. So these are the mouse chondrogenesis cells in vitro, which I showed you before. And she simply treated them with Sorfan, as I showed you before, or she knocked out EXT by adenocre. In all these cases, she got about usually it's 1 or 2-fold increase in mRNAs, gene expression, both RNA and protein level. Okay, so again, you lose heparan sulfate, actually the cells respond by making heparanase. Okay. So this is my last slide. Well, one more, just the acknowledgement. So this is roughly where we are moving toward. So we think that the formation of S-cystosis is caused by these three major changes. First, because you are losing heparan sulfate, there is an unrestricted distribution and overactivity of signaling factors, mostly prochondrogenic factors, BMPs and hedgehogs, as I showed you before. The second is that because the cells are HS deficient, actually they become even more responsive to the BMP and hedgehog. And third, I showed you before, this has to be confirmed in the patients, that there might be a positive prochondrogenic loop involving endogenous heparanase. So if you could think about these three combined systems, really lower as much as possible, they control the amount of heparan sulfate and really increase dramatically probably the responsiveness of the cells to these factors, and therefore inducing ectopic chondrogenesis and S-cystosis formation. Okay, so this is Children's Hospital in Philadelphia at nighttime. Most of what I showed you was done by Julian Hugo, who was a graduate student, Federica Sgariglia, who was a postdoc, with my wonderful colleagues, and our chief, John Dormant. Almost everything I've showed you was done in collaboration with Jeff Esco at USSD and Yu Yamaguchi here. We thank the NIAMS for giving us a good amount of money to do all this. Thank you so much.
We have time for probably a couple of questions. Jeff?
I guess convincing.
Maurizio, like when you look at surgical specimens or you look at the knockout mice, if you stain for the levels of heparan sulfate in the tissues, they're much more dramatically affected than what you would predict based on cell culture experiments where EXT is, where one copy of EXT is deleted. So I just wonder, in fact, if your induction of heparanase now is partially explaining that over-accentuation of loss of HS in the growth plate regions.
Yes, so that's why we followed up that study, because it would offer an additional possibility of why is the tissue, why are the cells so deficient of heparan sulfate, and why are they so responsive to this? Yes, so that would offer, yeah.
Culture systems where you can upregulate this.
Yes, so as soon as once we confirm the human data and we confirm also the mouse data, then we were going to go ahead and do the double transgenic mouse, because the heparanase knockout mouse has essentially no phenotype, which is great. And so we're just going to combine. If that enzyme has a role, then the mice should be more resistant to induction of S-cystosis formation. If that's the case, then we have a genetic proof that in fact this gene might be necessary for induction of S-cystosis. So but we're not going to do it unless we are sure that it might work. Yes. Yeah. So this is extremely controversial in this field. So a few patients have been found to be very